Bismillahirrahmanirrahim. The process of activation of target cell receptor by insulin is discussed in this video. The insulin receptor is a membrane receptor protein that has got the molecular weight of about 300,000. It is composed of four subunits, two alpha subunits and two beta subunits that are linked by disulfide bonds. The alpha subunits are located outside the cell extracellularly, outside the cell membrane, and they are responsible for the binding of the insulin, while two beta subunits, they are transmembrane proteins. They penetrate the cell membrane and extend into the cytoplasm. Insulin binds to the alpha subunit that are present in the outside the cell membrane, and this triggers autophosphorylation of the beta subunit inside the cell. This receptor acts as enzyme-linked receptor. Then autophosphorylation activates a local tyrosine kinase enzyme. This tyrosine kinase enzyme phosphorylates other intracellular proteins and activates IRS or insulin receptor substrates, for example, IRS1, IRS2, or IRS3. Different tissue express different types of IRS or insulin receptor substrates. This leads to activation and inactivation of multiple enzymes. So, this receptor activation will lead to various effects in the cell. There is a promotion of the glucose uptake and glycogen synthesis. Then there is enhanced fat synthesis and storage that and also reduced lipolysis. Then protein metabolism is this enzyme system, this activation of these receptors and enzymes that stimulate protein synthesis and inhibited the breakdown of the protein also, and also responsible for growth and gene expression. There are some rapid effects that occur within seconds, and within seconds about 80% of the body cell, especially the muscle and adipose tissue, show a rapid increase in glucose uptake, and after the insulin binds with this receptor. Intracellular vesicles that contain glucose, that contain glucose transporter or GLUT4, they move to the cell membrane. They are translocated to the cell membrane. These transporters facilitate glucose entry into the cell. Once insulin is gone into the cell, uh, these vesicles are removed from the membrane in 3 to 5 minutes and recycled. Immediately after entering the cell, the glucose is phosphorylated, trapping it inside the cell and making it available for carbohydrate metabolism. Then there is increased uptake of other molecules, and the cell membrane become more permeable to amino acid for protein synthesis, potassium ion, phosphate ion. And there are some intermediate effect that occur within 10 to 15 minutes, and these are enzyme regulations, and insulin alters the activity of multiple metabolic enzymes primarily via phosphorylation and dephosphorylation. Then there is a long-term effect that occurs from hours to days, and it is through gene and protein regulation. This is translation of messenger RNA that occurs at ribosome is altered to produce new proteins, and transcription of DNA in the nucleus is also modified. This remodels the enzymatic machinery of the cell, leading to the sustained metabolic effect. So, insulin exerts its effect at multiple time scale. Within seconds, there is transport of the glucose into the cell. Within minutes, there is enzyme activation, and within hours to days, there is gene expression and protein synthesis. To summarize these events, we can say that insulin binding to its receptor triggers tyrosine kinase activation via autophosphorylation of the receptor. This activates IRS protein, that are insulin receptor substrates protein, which function as signaling hub, leading to multiple metabolic outcomes like glucose uptake, fat synthesis, protein synthesis, and gene regulation. Thank you for watching this video. If you have any query, please write in the comment box.